Hello everyone, we're here today to talk about the estrous cycle. Well, I'm hoping those of you watching this have a basic familiarity with what the estrous cycle actually is. I'll give you a definition to clear up any confusion. The estrous cycle is, and I'm taking this word for word from the Pathways to Pregnancy and Parturition textbook, a series of predictable reproductive events beginning at estrus or heat and ending at the subsequent estrus. So, why do we care? The keyword in the previous definition was predictable. If you know the estrous cycle and can understand hormone levels, physiological events, behavioral signals, and ovarian events, and how those all relate, you can manipulate the cycle for assisted reproductive technologies. This has been key in improving the genetics of our production animals over the years and is super important so we don't waste valuable resources by, for example, inseminating a mare who is in a stage of her cycle where there's no possible way she could conceive. Understanding the estrous cycle beyond just memorizing what it looks like is absolutely key to understanding reproductive physiology. So, without further ado, let's jump in. Now, this diagram right here is a drawing of a cow's estrous cycle. It was taken from the University of Penn's vet school, and I added a line for inhibin as I was unable to find a good picture of a estrous cycle that also included the hormone inhibin. So I know it looks pretty complicated at first, but I promise we'll break it down so it makes more sense. First, we'll look at each hormone, what they stand for, and some of their functions. I just want to warn you now, we'll be discussing these hormones in the order of the hypothalamal-pituitary-gonadal axis for reasons that will hopefully make sense as we go along. If you're unfamiliar with this term, don't worry. There will be an overarching explanation at the end after you've been introduced to each hormone in order to discuss their interactions with each other. So the first hormone we're going to discuss is actually not on the last diagram, but it essentially starts the cycle and is super important, so I think we should mention a little bit about it first. This hormone is GnRH. GnRH stands for gonadotropin releasing hormone. As its name implies, it controls the release of the gonadotropins, FSH and LH, which will be spoken about in a moment. GnRH is released by the hypothalamus, particularly the tonic center, and after certain events discussed later, the surge center. It targets the anterior lobe of the pituitary and tells it to synthesize and secrete the gonadotropins. The frequency and amplitude of secretion of GnRH has an effect on which gonadotropin is secreted in higher amounts, which will be important in making sense of the graph that was shown in the last slide at the end of the video. But for now, let's move on to the gonadotropins. So here is where we should think about our Latin roots. Troph means something nourishing, so the gonadotropins in general nourish the gonads. So if you're ever stuck, remember that gonadotropins are something that promote gonadal function. Luckily, their names are fairly self-explanatory, as they were named specifically for what they do in the female. So FSH is follicle stimulating hormone. So, in the estrous cycle, FSH causes follicles to grow and mature. FSH is produced in the anterior lobe of the pituitary and targets specifically the granulosa cells of the ovary. As it stimulates the follicle to mature, it also induces estradiol and inhibin synthesis. The more the follicles grow, the more estradiol produced. LH, the other gonadotropin, is luteinizing hormone. A spike in LH stimulates ovulation and the formation of a corpus luteum, aka luteinization. The corpus luteum is what will produce progesterone during luteal phase, which will be discussed later in this video. LH is produced by the anterior lobe of the pituitary and targets the theca interna cells, and later the luteal cells, which are formed by both thecal cells and granulosa cells, after ovulation. Okay, so now that we've discussed the gonadotropins, we can move on to the actual gonadal hormones. So E2, while I realize it's not labeled like this in this diagram, is a common abbreviation in your textbook for estradiol. Estradiol is a steroid hormone secreted by the granulosa cells of the growing follicles in the ovary. It is the primary hormone of follicular phase, which will be discussed a little later in this video. Estradiol acts on the brain in both males and females in order to stimulate reproductive behavior, for example, lordosis, or the arched back posture indicating that a female will allow a male to mount her. Estradiol also enhances the motility of the uterus and increases the secretory activity of the female reproductive tract. Inhibin is a glycoprotein hormone that is also secreted by the granulosa cells of the growing follicles. As you might imagine, inhibin is an inhibitory hormone. Specifically, it acts on the anterior lobe of the pituitary in order to inhibit FSH secretion. P4, which once again is an abbreviation not used on this diagram but very frequently used in your textbook, stands for progesterone. This steroid hormone is produced by the corpus luteum and it is, in general, very inhibitory. Progesterone inhibits GnRH release, inhibits reproductive behavior, and reduces myometrial contractions, except in the mare. However, this is the hormone that promotes pregnancy, so it stimulates alveolar development in the mammary gland and secretion from the endometrium of the uterus. Okay, so now that you've gotten the basics of the main hormones at play in the estrous cycle, I'll explain why everything was explained in this order. This is where the hypothalamo-pituitary gonadal axis comes in handy. So, for a reminder, this is essentially the order in which I talked about these hormones. Note that these can be split up by where they were produced. This is why it's called the hypothalamo-pituitary gonadal axis. So essentially, the axis is what I drew here, but with the negative feedback and positive feedback relations drawn out. It describes how sensory information, or central nervous input, or simple tonic secretion in the case of GnRH, starts up from the hypothalamus, which is the master gland of the endocrine system. It describes how the hypothalamus starts a chain of different hormones releases that results in the physiological effects we can observe. Hopefully, having this information in a handy graph format will help you remember those hormone interactions, which can be a bit difficult. So let's get to work fleshing it out a bit. Let's start with inhibin, as its function is simply negative feedback on FSH. You can think of this as a neat little thermostat type control. FSH means growing follicles, which means more inhibin, which means FSH is reduced. These follicles will still grow due to growing receptors for other hormones to help them grow, but that is a subject for later. So the only hormone inhibin really affects is FSH. So, moving on, estradiol is one that you guys should have discussed before, so hopefully this is review. Estradiol is like inhibin and is inhibitory on FSH secretion. I like to remember this by remembering that these guys are being produced by the same cells in response to the same stimuli, aka FSH secretion, so it is likely that at least part of their function is the same. However, this isn't the full story with estradiol. Estradiol has a different relationship with the other gonadotropin, LH, as well as GnRH. When at low concentrations, estradiol has negative feedback on LH and GnRH. This negative feedback is somewhat of a biological norm. When you think about it, it makes sense. When you start producing something, it's logical for the product to inhibit the process, or you could end up with too much product and no way to stop its production, and excess usually isn't a good thing in biology. However, when estradiol reaches a certain threshold concentration, it activates the surge center of the hypothalamus and begins positive feedback on GnRH and LH. This LH surge results in ovulation, so it's very important. Finally, let's talk about progesterone. I'm going to switch to another color here because I realize it's getting a little cramped. Unfortunately, there's not as much room on an iPad as there is on a whiteboard. So I saved progesterone for last as it can be a bit complicated, so I promise I'll try to simplify it as much as I can. So I mentioned earlier that this was a highly inhibitory hormone and that in particular it inhibits GnRH release. Because GnRH is the beginning of this pathway, then you can infer it would decrease levels of circulating FSH, LH, estradiol, and inhibin. It does also have a direct inhibitory effect on LH. Sorry about that very complicated line. So when progesterone is dominant, you tend to see very low estradiol, very low LH, and very low GnRH. However, and this goes back to what I mentioned earlier when discussing GnRH and how different pulse levels favor different gonadotropins. So FSH is actually favored by slow GnRH pulses, so it's not affected by progesterone dominance like the other hormones discussed here are. So progesterone can go and inhibit GnRH, but GnRH is always produced. So these slow pulses just means FSH can still grow. So this information will help you be able to make sense out of some of the later FSH peaks that we'll see in the cycle, which are all due to follicular dynamics, which is a subject for a whole different video. So now that you're familiar with the hormones and their interactions with each other, let's go into the different phases and different stages of the estrous cycle and why you would see differing hormone levels at these points in time. There are two big phases that you should be familiar with, and these are the follicular phase and the luteal phase. The follicular phase can be split up into two stages, proestrus and estrus. Similarly, luteal phase can be split into medestrus and diestrus. Now, we'll look at each of these stages in slightly closer detail. In follicular phase, which makes up only about 20% of the cycle and lasts for about 5 to 6 days in cows, a female animal is building up her follicles in preparation for ovulation. In proestrus, follicles are growing and being recruited and selected towards dominance. Now, I just want to say the reason why I have two graphs in here, one from the pregnancy and parturition textbook, is because I think it gives a really good idea of how to split up the different stages of parturition. However, the University of Penn diagram gives more hormones for you to look at. So hopefully looking at both these will help you guys make sense of everything. So, going back to proestrus, we can look at our different hormone levels during those stages. As we mentioned, follicles are growing and being recruited, so FSH should have just finished peaking, and estradiol and inhibin are increasing as the follicles grow and get increasing steroidogenic capability. Progesterone is low because there's no corpus luteum in the ovary right now, so progesterone should not really even be able to be produced. LH remains at tonic levels because estradiol is not yet built up high enough to reach that threshold value that stimulates the positive feedback on GnRH and LH. Alright, so now that we've discussed proestrus, we can move on to estrus. In estrus, the follicles are getting really big at this point, and the animal's starting to become sexually receptive. This is where you hear that term in heat. If she is in heat and thus demonstrating reproductive behavior, you can bet that her estradiol is increasing and peaking. Because estradiol is peaking and has passed the threshold concentration, we have an LH surge, which in most animals is a fairly sharp peak right before ovulation. The big follicles mentioned earlier mean that inhibin was produced along with estradiol. And now that we've got large amounts of estradiol and inhibin, FSH is probably going to start declining. And just to connect the whole big picture, physiologically, our huge amount of estradiol is increasing the secretions and motility of the female reproductive tract in preparation for mating in the sperm she hopes she'll be receiving close to ovulation. Ovulation marks the transition from follicular phase into luteal phase. In luteal phase, the female is preparing the body for pregnancy, which is the hoped-for outcome after ovulation. In meta-estrus, ovulation has just occurred, so the follicle has just been destroyed. Remember, the follicle produced estradiol and inhibin, so we're going to see a big decrease in estradiol and inhibin because the follicles that produce them have either undergone atresia before ovulation, or, in the case of the dominant follicle, have ovulated. LH remains at tonic levels due to a lack of estradiol to stimulate another surge. It is simply present in high enough amounts to stimulate the conversion of thecal and granulosa cells into luteal cells. Remember, luteinization hormone, so it's trying to form the corpus luteum. Progesterone is on the rise, but the luteal cells haven't been fully produced yet, so they haven't reached their peak steroidogenic capability, so peak secretion of progesterone has not yet been reached. And finally, we do have a small FSH peak. While we haven't really talked about follicular dynamics in the video, it does explain this peak. So remember that progesterone does not inhibit FSH, and that progesterone is not very high yet in meta-estrus in any case. So FSH can be secreted and follicles do start to grow. I'll explain the fate of the follicles that have started growing because of this FSH peak once we get into diestrus. Now, the main event in diestrus is that we're reaching our peak in progesterone. This progesterone, secreted by the corpus luteum, which is now the main structure of the ovary, will remain high until luteolysis around day 16, which I guess you can kind of say is within proestrus, unless the animal becomes pregnant, in which case the progesterone will maintain high throughout pregnancy. And, just as a reminder, remember the physiological purpose of this progesterone is to prepare the animal for pregnancy. So, reduced contractions in the myometrium, secretions of the endometrium, increased mammary gland development, decreased reproductive behavior since she's already hopefully been mated, etc. Now, as progesterone is inhibitory on GnRH, LH will remain at low tonic levels. Now, this is what's important for those follicles that I mentioned earlier. That small peak in FSH led to a small peak in estradiol and inhibin, as we've discussed multiple times throughout this video. However, even though estradiol may be increasing, progesterone has a hugely inhibitory effect on LH, as we just mentioned. So, even if estradiol reached a peak level, it cannot stimulate an LH surge, meaning it cannot stimulate ovulation. Therefore, all of the follicles from this first wave of recruitment will all go into atresia. This clears the way for second wave recruitment during diestrus, which are the follicles that will eventually ovulate in estrus. So, therefore, we have another peak of FSH and another peak of estradiol. Alright, so that's been a lot of information, and hopefully now we can all put it together and really understand this estrous cycle. So we're going to take a look at a blank cycle and work our way through it. So I know there was just a lot of information dumped on you at once, so hopefully this last review is what makes it clear one more time. So first of all, we should remember that the estrous cycle, day 0, is always ovulation. And the cow's cycle is about 21 days on average, so I'll draw enough room for one full estrous cycle, plus a little room on the side so that you guys can see the pattern. So let's have ovulation right here. And we'll have 0. And I'll pause for numbering to save you guys some time. Okay, so now that we've got our basic graph, I tend to just go through with each hormone and try to remember, okay, what should it be looking like at various phases of the cycle. So I always start with the LH peak because it just peaks right before ovulation, and then it's just tonic secretion the rest of the time. So here we can draw LH, and I'm going to choose yellow as that seems to be the color I see it in a lot. So here are 2 ovulation peaks, so tonic secretion, and then we've got a peak right before ovulation. Tonic secretion, peak right before ovulation. And that is LH. So I find progesterone to be another really easy peak to draw because we know it only can peak after ovulation, after the corpus luteum is formed. So let me change my color to something a little darker. Okay, and so with progesterone, I can draw a slope up starting after ovulation. And if we remember, med-estrus was about 5 days, and progesterone peaks at the beginning of diestrus and remains high. So we can draw a little peak starting at ovulation that goes up to about day 5. And now we know it's going to remain high until that corpus luteum is destroyed. And I'm not sure if I made this super clear, but in case I did not before, luteolysis happens at about day 16. So I can keep this peak high until about day 16, and then we get luteolysis, and it plunges. And it'll remain low until we get our ovulation again, and then we get another increase. And then I'll just extend this past here. So there we go. So that's our P4. And I like to draw FSH then next, as I know there can be follicular waves that don't ovulate and follicular waves that do. So there can be a wave when progesterone is on the rise, and that was in med-estrus, which we just talked about. And that's the one that doesn't ovulate. So it's just going to come up. Let's see, med-estrus is right after ovulation. So we've got a little peak in med-estrus. And, however, we do know that there's going to have to be a follicular wave that does ovulate. And for it to grow and grow enough, for these follicles to grow enough for us to have it ovulate at estrus, it needs to start in diestrus, probably around day 10. So this is where we're going to get a growth of follicles. And there also can be in cows a third follicular wave, and this tends to happen around over in our proestrus-estrus area, I believe. So there's our third little peak. So there's our FSH. And that can be the end of that peak there. So here's FSH. Okay. And now that we know our FSH curves, we can draw our inhibin curve, because inhibin is released due to stimulation by FSH, which then inhibits its secretion. So it makes sense for the inhibin curve to look like the FSH curve, but slightly after it. So I'm just going to take some blue, and I'm going to follow my FSH curve, but I'm going to have it drop just a little bit after and rise a little bit after. And that can be my inhibin curve. There we go. Excuse me, sorry about that. Okay, and now finally, we need to do estradiol. And estradiol is also released due to stimulation by FSH, so its curve will also begin slightly after FSH. It'll have a similar curve for both the non-ovulating and ovulating wave. But remember that the increase near ovulation is exponential because it's triggering that LH surge. So now I'm going to go pick a color for estradiol. Let's go with green. We haven't used green yet. So this is going to get a little messy. So here's our estradiol. You know it's peaking. It's very similar to inhibin. So let's draw it by inhibin, by inhibin, by inhibin, And then we have our huge surge here that's causing the LH surge, and then it's dropping after ovulation. So there is our E2. Okay, so this takes us back full circle, and we have a nice graph with all 5 of the hormones that we've been discussing, and that, ends our discussion. So I hope this video was able to clarify some of the more difficult aspects of the estrous cycle. So please leave a comment below if there's something we missed or if there's some other aspect of this you'd like us to cover.